If I was to take a plant, let's just pretend the plant has multiple Lego blocks that are required to make the plant, and you're not given the instructions to make this plant. Let's say you have another plant as well, and that also has its own building blocks. You're given some parts of the manual. You notice that for the one plant that you are given the manual for the building blocks, there's similar pieces that are in the one that does not have the manual. So you're noticing similar building blocks, and you want to see, okay, well, I have millions of building blocks to construct these plants. How can I compare them? We are also looking at how these two building blocks can be destroyed or affected by adding different stimuli.
We had one species called Arabidopsis thaliana. This is a mustard plant. It's a typical model for plant biologists. And another type of plant, Brachypodium vistachyum, which is a grass species. So the two plants are pretty different. But what we did is we extracted their cells, kind of pooled them into a single tube, and then profiled them at the same time.
So we're essentially adding to a giant database, and we're looking at those reactions, and we're ensuring that, hey, maybe we shouldn't do this to this plant, or maybe we should.
In any kind of discovery-based project, you never know exactly where you're going to end up. It always kind of is a dynamic, evolving process.
It's harder to compare Arabidopsis with Brachypodium, and we really had to just take a complete new direction. It's still great. It's just not the initial motive that I remember hearing in the beginning, but that's not a bad thing. Ben actually taught me a new way of thinking. I was able to learn how to more iteratively look at things, and that is actually one of the most essential things to know for data science. So I'm definitely going to take that process back with me.